this A-level biology revision video with Mr. Phillips, and today we'll be looking at the eukaryotic gene expression control using something called transcription factors. Now, I think the picture at the top of this page is really useful for us to start off with this. A transcription factor is basically something that allows the attachment of RNA polymerase to the area of the gene in the DNA, which allows transcription to begin. So a transcription factor might be a protein. It attaches to something called the promoter region, which isn't the bit of DNA that is being used to make the protein, but it's next to that piece of DNA, and therefore it allows RNA polymerase to attach, and then RNA polymerase can do transcription, and then obviously that makes the messenger RNA in protein synthesis. By gene expression, we talked about this last time in the last video. It's about the genes being switched on effectively. And we can alter whether a gene is switched on by stopping it from doing transcription. So only some genes get transcribed into messenger RNA, or we can destroy the messenger RNA, preventing translation or blocking it at the ribosome, preventing the polypeptide from being made. So how does this work? What is a transcriptional factor? How does it work? Well, basically all DNA base sequences for every gene, I should say, has a DNA base sequence of about 100 base pairs, this green bit here called the promoter region. Now, this is essential because this is where the RNA polymerase has to attach to make the messenger RNA for that gene. So this orange bit here represents the bit of the gene you're actually going to use to make the protein with, the functional gene, or we could call it the exon. And this bit then is the promoter region where the RNA polymerase attaches. But in order for that RNA to polymerase attach, we need something called a transcription factor. Is a molecule that attaches to this and allows RNA polymerase to then bind, and therefore allows transcription. So if you don't have this transcription factor here, you won't be able to have messenger RNA, because the RNA polymerase will not be able to attach, and then it can't move down the functional gene to make the protein. So let's have a look at how this works. When the gene is switched on, a transcription factor, which is a specific molecule that attaches to the promoter region, is present, and it moves from the cytoplasm into the nucleus. Each transcription factor or transcriptional factor has a binding site specific to the sequence of DNA at the promoter region. So hence it's specific to that gene. So that promoter region will have a certain code, and the transcription factor will have a complementary code. When it binds to this, it allows RNA polymerase to bind, and that then allows transcription of that gene. RNA polymerase is then spliced, which we'll remind you about at the end of the video, and then translation occurs. If the gene is not expressed, however, the binding site is not active, and it can't therefore bind, the transcription factor cannot then bind to the DNA, and therefore your RNA polymerase cannot bind and factors get activated, which means that they therefore go into and onto the promoter region, allowing that gene to be transcribed and making that protein, because we obviously only want these to happen when we need them. Well, often they're activated by another chemical. So an example chemical we're going to use today is the hormone estrogen, which is a steroid hormone, which means it's lipid soluble. It can go through the plasma membrane. Estrogen we associate obviously with the menstrual cycle in females, but actually estrogen is an important hormone in males as well, because it actually initiates sperm production. And obviously the menstrual cycle in females. And we're going to be talking about estrogen initiating the activating the transcription factor ERA. So that is a transcription factor which is in a protein complex in your nucleuses. So if we look at this, then what happens is estrogen comes in. Estrogen is a steroid hormone. It is lipid soluble, so it will just diffuse straight through the cell plasma membrane. So you don't have a first and second messenger as you would with, say, insulin here, or any G proteins or anything like that. It just goes straight through into the cytoplasm. That then diffuses into the nucleus, because that again is where your genes are. Okay, and then when it goes into the nucleus, it attaches to a protein complex and binds with this ERA, which is a transcriptional factor. And it's a transcription factor for hundreds of different genes. Now, once that has binded to that, that activates the EAR, which basically means it changes its shape, so it does a conformational change, and that then leaves the protein complex, and then it will bind to the promoter region, which allows RNA polymerase to attach and to start transcription. So if you look at it, the lipid, the estrogen comes in, it binds to a receptor on the ERA complex that alters the shape through conformational change of the ERA. The ERA then can now attach to the promoter region, and that then causes the polymerase to bind, and that then allows RNA polymerase to start transcription. We can use our knowledge of transcriptional factors to treat potentially diseases. And one of the key types of disease that is linked to estrogen levels is breast cancer. So 35% of breast cancers are caused by estrogen overstimulating transcription factors, that then leading to uncontrolled cell division. And there are different types of drugs that have been used. So one type of drug is tamoxifen, which is similar enough to bind to the estrogen receptor, but does not change its shape. So effectively it blocks the estrogen receptor, meaning that the ERA is no longer activated, and therefore it won't create it won't allow the RNA polymerase to attach, and therefore stop excessive transcription. There's a new drug called aromatase, which is also used to inhibit the enzymes involved in estrogen synthesis as well, so you can prevent that from happening. Obviously, some cancers and tumours are not estrogen linked, so an example of one way of treating these could be something called Herceptin, and Herceptin is a monoclonal antibody complex which binds to growth hormones in the plasma membrane of cancer cells, and it stops their division. So there's lots of different ways that we treat cancer, and this will be used in conjunction with chemotherapy. The problem obviously with these drugs is they're extremely expensive, and that limits the people that can be treated. Really it for our transcriptional factors, but just to remember that actually the gene regulation can happen post-transcriptional, and you would have talked about this in the first year when you've talked about splicing, and you know that splicing, when you have your original DNA, there are introns and exons. So introns are non-coding regions of DNA, it's ones that don't code for polypeptides, whereas exons do code for polypeptides, and you make primary RNA from transcription before they are the introns are then removed from the RNA, and they do this by cutting out the exons, introns, before combining the exons together using an endonuclease enzyme. And splicing method can actually be used and manipulated to cut out different genes, and therefore more than one type of protein can be made from the same DNA. We can actually activate transcription factors. So estrogen obviously activated that one earlier on. And another example where you might link it in the course is something called phosphorylation. So when we phosphorylate something, where we add a phosphate to a molecule, it can activate transcription factors, therefore changing their shape and allowing them to attach to the promoter region of the DNA. An example of this is using the enzyme galactokinase,